Welcome back to this conference, and I'm very happy to greet not only you, but our first speaker who arrived yesterday evening, who is not unknown to NTU. Professor Paul Matthews is our honorary visiting professor at LKC Medicine, and more, he is the head of the department of the Division of Brain Sciences, or head of the Division of Brain Sciences at Imperial College, which is our partner institution regarding our medical school, LKC Medicine. As you may know, LKC Medicine is a joint venture between NTU and Imperial College London. Paul is one of those rare animals in neuroscience who have a PhD, more precisely a DPhil, from Oxford in biochemistry. So he started as a biochemist under Professor Sir George Rudder, chairman of Singapore's Biomedical Research Council, at that time professor of biochemistry at Oxford University. He then went on with his medical studies at Stanford University, and after obtaining his medical degree, he went to the world-famous Montreal Institute of Neurology and made his specialization in neurology in Montreal. Then he returned to the UK, where after many different clinical and research positions, he became vice president of GSK, responsible for research drug development. And after the establishment of the medical school at Imperial College, he became the first head of the Division of Brain Sciences. So Paul, welcome. The word is yours.
So Balash, thank you for the invitation to join you here today. I'm genuinely looking forward to the next 2 days to learning a great deal. It's an incredible assembly of people, and it's exciting to be back in Singapore to see what you've built. I come here regularly, but not frequently, about twice a year. And I must say twice a year is intermittent enough that big changes occur, and it's always exciting to see what happens between each visit. And I know we'll see something this week that's new to me. So what I'm going to try to speak about today is a general challenge for brain sciences as we start to think about, particularly about interdisciplinary activities at the interface with medicine, or even more challenging as we're taking on over this 3 days at the interface with the arts and understanding who it is that we are as a species. I'm going to take up a much more limited problem, which is really how we can begin to understand the relationships between individual variation in brain functional architecture at a population level to begin to make some of the associations which might ultimately lead to causal explanations for elements of this variation. The problem of understanding who we are is one that has, of course, has taxed most of the Renaissance, and in this era has been instantiated very much in terms of trying to understand a brain basis for who we are. This has become very much part of our popular culture, as illustrated by this quote from James Gorman, one of the columnists for the New York Times, after his visit to the Human Connectome Project main campus at Washington University about a year and a half ago. I was looking for a hint of my own individuality. So it motivated him to go through the HCP protocol and to, with some excitement, to try to hunt for that within the structural and functional basis of his brain that was described. Now, looking for our individual variability is at a population level has been going on for really since the earliest days of imaging, and if not before. So the modern era, I think, was heralded by Danny Weinberger's seminal studies attempting to understand, as part of an attempt to understand the basis for expression of psychiatric morbidities. What he did is began one of the first population-based imaging studies. At his time, it was limited to some tens of subjects, and he used reconstructions of surface structure of the brain, which you can see here illustrations from a post-mortem example that he was correlating with his imaging studies, in which he made some very important simple observations which remain very much the case today. The first is that gyration patterns in the brain are highly variable. They are almost as variable as our fingerprints. And if one looks across individuals, you see very different patterns of folding in regions of brain, particularly in prefrontal and temporal regions, as illustrated here. But if one looks at people who share a common genetic background, here monozygotic twins, one sees really remarkable preservation of major features shown in the frontal operculum here, relative to those who share fewer alleles, in this case dizygotic twins. So what this illustrates is that there is a tremendous amount of surface structural variation in the brain, the mechanisms for which are still uncertain, and that these are heritable to some significant degree. Now, as this study was extended further to a larger population using a mapping technique which helped the investigators express surface structure in terms of measures of common curvature across a simple matrix that was defined common to all brains. What became clear is that while the heritability of features was present, it was actually rather modest. And you can see here between unrelated and twin individuals, its surface structure is only slightly heritable. Now, the next advance came about a decade later with work coming out of the UCLA lab of Arthur Toga and Paul Thompson, where they expanded the population study and took a finer-grained look at the problem by mapping not just surface gyration, but starting to look at surface microstructure through measures of that are surrogates for cortical thickness and cell packing, so-called cortical density measures. Now, we don't need to worry too much about the correspondence between the imaging-derived features and the underlying cellular basis at this point. It is approximate only, but this measure gives rise to an increasingly clear pattern, and that again expresses the notion that there is considerable variation in cortical microarchitecture across individuals in a population. This is an arbitrarily thresholded variance map of the cortical density voxel by voxel across the brain, and that the degree to which features are shared in common regions of brain space rises with the degree of genetic relatedness. So here in identical twins, monozygotic twins, we see that there is a highly significant degree of heritability of features, more similar being pink, relative to other in some regions of brain. Now, I think what's more interesting to me than the general point that these features are under a degree of these features show a degree of heritability is that the heritability is spatially varying across the brain in ways that we'll talk about throughout this talk. And in general, in the prefrontal cortex, particularly the inferior frontal area and the temporal pole, extending more posteriorly in the temporal lobe, there is a remarkably high degree of variance between individuals that does not seem to be explained by this common allelic structure. Now, I've talked about the surface gyration. I've talked about some aspects of cortical microstructure as reflected in imaging. Moving deeper to that, of course, there is a connectional architecture related to the long-distance projections in the brain. At the level of human clinical imaging, we're looking at a rather crude surrogate of this, which is a diffusion MRI measure of orientation of fiber bundles and relative packing. And these can be expressed in a variety of ways. What you see here is a skeletonized version of this Milo architecture, in which the local fiber tracts have been collapsed down to a backbone. The reason for this is because it allows much more precise registration across individuals at the cost of some anatomical resolution. This particular map, developed by Kochanov and his colleagues, looks across a population including, again, monozygotic, dizygotic twins, and unrelated subjects in order to map a heritability index to the large fiber bundles as collapsed here and expressed as a shared fractional anisotropy or measure of fiber orientation. Now, what one sees is that the areas in yellow-green are those of higher heritability index, 1 being bright red, 0.2 being blue, and one sees as one proceeds towards central large projection tracts of the corpus callosum, looking at this in sagittal section, and the deep projection tracts going down the brain stem, that there is a greater heritability to the architecture of these bundles, which means they're packing they're packing and relative orientation in the common brain space. As we move out towards the cortex, particularly anteriorly again into the prefrontal regions, one sees lower degrees of heritability. Now, what is remarkable, and one has to take this as somewhat sample and certainly technique dependent, what is remarkable is beginning to probe the heritability of specific tracts plucked from this compressed summary version that I've shown you over here. What is described here in this rather beautiful plastic molding are individual tracts segmented out from the diffusion imaging. Here, for example, you see the posterior limb of the internal capsule as it moves down. Here is the external capsule. Here is the anterior limb of the internal capsule going down, and so on. Now, each of these has an architecture defined by its spatial extent, packing, and packing measures as reflected in the imaging that can be defined, can be tested for heritability. And what one sees here is really rather remarkably, there's an extraordinary degree of heritability in the population that was studied. We're up to measures approaching 0.9 in these bright red regions. Rather than the precise figure, however, is the way in which, again, this heritability varies spatially. And if you notice, in contrast to the very high heritability of these large projecting tracts going into the brainstem or extending posteriorly across the brain, you see here the fornix shows a much lower degree of heritability, and again, other areas such as the cingulate fibers also show distinct patterns. So there is, we can begin to use all of these features to try to understand something about individual variation in architecture of the brain. And of course, it's this architecture which does not independently determine, but which provides a context for the functional architecture of the brain. I expressed the notion at the beginning with some humility that we're looking at, with the conventional structural imaging measures, at surrogates for anatomical structure. The level of resolution, which is running in these types of experiments between 0.7 millimeters on an edge and 1.2 millimeters on an edge, is far, far lower than the size of the structures being measured. And of course, these measures of connectivity do not themselves express anything about connectivity as determined by the synaptic architecture. Now, to begin to get some understanding of synaptic architecture, we need to move to functional surrogates. I'll talk now about extending these kinds of measures to looking at shared functional architectures of the brain, which begin at least to sample some aspects of synaptic function. And one of the first, I think, really compelling ways of beginning to integrate these two kinds of thought came from work that we did now about a decade ago in Oxford, led by Heidi Johansen-Berg with Tim Behrens, in which there was an attempt to parcellate, in this case, a small section of the medial prefrontal cortex. We're looking now at a section of an axial image, illustrating the kind of resolution that was obtained from the diffusion tensor maps. And this is the midline cortex. This is the white matter. And the way in which this was done is the myelo-architecture was used in order to infer variation in cytoarchitectonics along the midline cortex. The logic behind this was very simple, and that is that different cytoarchitectonic regions in gray matter with their different projection architectures have distinct patterns of white matter connectivity. These can be mapped using a probabilistic tractography approach developed by Behrens, and one can begin to assign, after grouping across a connectional matrix, regions of distinct connectional architecture as determined by the long projecting white matter tracks. Here in the medial prefrontal cortex, one could define two regions. This was the area explored. Here, on reordering the matrix, one could see that there were two quite distinct clusters of connectivity, one which connected predominantly anteriorly to frontal cortex, one which projected much more diffusely and posteriorly. Hypothetically, these were assigned to the supplementary motor area and to the pre-SMA. And this assignment then was able to be validated by looking in the same subject. This is a single subject's examination at the functional landmarks defined in the same area by using a task-based functional magnetic resonance imaging experiment in which the subject was asked to move the fingers in guided succession, which activates the supplementary motor area, and separately to perform a serial subtraction test, which is a strong activator of the pre-SMA region. The regions of predominant activation shown here for the latter task in orange, for the former task in blue, overlapped very strongly with an almost direct border, slightly fuzzier than this that we see in the structural map with the structural correspondence we see there. So what that's beginning to do is is one of the earliest experiments that draws a direct correspondence between aspects of functional architecture and those of the structural architecture. Now, it was also about this time that Marcus Reichel made his really rather remarkable observation of of low frequency oscillatory behavior in the brain, a heavily damped, very low frequency set of oscillations that were occurring in the initial instance, predominantly in frontal parietal connectivities involving precuneate and prefrontal regions as illustrated here. This pattern of brain activity was remarkably well preserved across individuals. It was persisted at rest and was damped during focused task activity, regardless of the task activity, and has since been explored very widely. Some have called this the so-called default mode network, and I won't begin to describe the very broad range of correlates for this. What is important to recognize is that this this represents a network of function that is working in concert with other networks in the brain. One of the really most important observations from the beginning about this was the way in which its activity was anti-correlated with those of other networks in the brain. Now, this has led to a very broad range of generalizations, but what this is telling us is that is that within the functional architecture of the brain, there is a time-varying signal that is giving us some spatially useful information, differentiating different clusters of functionally related neural populations at the cortical level. Again, very large populations. Now, one example of this that extends the paradigm somewhat came from Apkarian's group at Northwestern, working with Marcel Messelam, in a really very elegant experiment in which they began to probe the relationship between local cortical blood oxygenation level dependent signal change, the fMRI signal change being observed by Reichel and others, and the long distance correlations in that signal between different brain regions, the so-called functional connectivity mapping. Let me take you through this a little bit slowly because it introduces some important concepts. The first concept is that within any region of brain, one can define, and I'll talk about this in more formal detail later, clusters of neurons that show highly correlated local time-dependent behavior. Again, the power spectrum for this is heavily weighted towards very low frequencies, as you see here in the log power distribution for that region. From this, one can use any of a number of spatial temporal decomposition techniques to define meaningful regions of similar variation. And here you see that one of the An example of one of these, and indeed one of the strongest ones, is pulling out exactly the same sort of frontal-parietal default mode network that Reichel spoke of some years earlier. What this highlights is that regions of brain show in their local power spectra with bold, both correlated and anti-correlated in blue regions of activity, as I was describing. Now, this is referring entirely to the local connectional architecture, or what's happening with observations only on a voxel-wise basis across the cortex. Now, if one extends this to begin to look at the correlations at long distance, making the diagonal elements of the matrix, ignoring the diagonal elements of the cross-correlation matrix, one can derive a different sort of map of correlation. And this is a map of correlations on a voxel cluster to voxel cluster wise level at long distance. And what's really important is that when they're filtered for similar powers, treating the bold as a low-pass filter, the patterns of activity become remarkably similar. Now, the significance of this is that this begins to draw a direct correspondence between the local signal properties, that is to say, the local activity in cortical regions and their long-distance relations. Now, in some respects, this is not surprising because we're defining the long-distance relations on the basis of correlations in the first place. But I want to highlight this because it makes the approaches to these connectional problems that have been used across the functional imaging literature much more transparently related. Now, I spoke about Reichel's discovery of the frontal parietal connectivities from analysis of his then extant functional imaging data. In fact, we were one of the groups to emphasize very soon after that these are much more widespread. And in Mara De Luca's paper from 2006, using a temporal spatial non-Gaussian ICA method as a data reduction approach, we demonstrated that one could reliably pull out from individuals the same set of 6 to 10 common connectional correlations across regions of brain. And moreover, that these tended to correspond to regions that were associated with functional architecture appreciated well from task-related fMRI activities, those associated with so-called attentional responses with executive, sensory motor, auditory, and visual perception or control. And so this gave rise to this and related work gave rise to the notion that these long-distance correlations between large clusters of coherent neuronal activity were related to an underlying functional architecture that was maintained for perception and action. For some years, the potential confound of the role of the neurovascular coupling unit in giving rise to these signals was a subject of hot debate. And it's worth mentioning this, if only parenthetically, because it has been so important to the conception of these areas and actually needs to be reconsidered now as we start to make finer and finer interpretations of them. Of course, the blood oxygenation level dependent signal is not detecting neuronal activity, but is detecting a complex consequence of local blood flow changes in the context of oxygen utilization with increased neuronal activity. Now, the correspondence, therefore, is indirect. In fact, despite this, it seems to be remarkably well preserved across the brain. And so in the healthy brain, correspondences between correlations observed at an electrophysiological level and those observed with functional imaging still seem to have held. I don't have time to describe these. These are not entirely straightforward, and they certainly vary with brain region, as one would expect by the different input structures of neurons. But one of the summary descriptions of this sort of phenomena came from Peter Morris's lab in a really rather elegant MEG study in which a group of individuals was studied using MEG with beamformer methods and fMRI, and similar low-pass filtered MEG patterns were derived and assessed for the spatial temporal correlations as the fMRI. And you see that using similar decomposition techniques with the electrophysiological or the bold imaging methodology, one can pull out very similar patterns of connectivity, at least at this broad level of abstraction between the two. This gives one more confidence that, along with other evidence, that these data are reflecting something that is related to the way the brain functions rather than some very indirect measure of this. Now, this pattern of logic together, genes to structure to physiology and expressed as function underlying behavior, has led to the real excitement in the area, which is the notion that one can begin to use the functional architecture as a probe either for changes in disease state, antecedents, functional antecedents of disease or risks of future disease, as well as possibly trying to understand the basis for individual differences in behavioral tree or state. One such example, although again very crude, is shown here from Ed Bowmore's work, where he's used a graph topological relationship with defined cortical functional and structural seeds in order to try to explore differences between the brains of people with schizophrenia and matched healthy control subjects, and at least in this first approach based on meta-analyses of a number of small studies, there is a suggestion that there may be consistent differences in the population of those with schizophrenia relative to healthy controls that may affect connectivities with a topology affecting particularly central white matter paths. Now, I don't want to highlight that as a conclusion yet, but as a direction of travel. And I want to use this also to step back and reflect for a moment on why many of us are becoming much more interested in the resting state as a way of understanding brain variation than in probing more conventional functional architectural expressions using task fMRI. And I think this concept can be expressed in many ways, but one of the most persuasive for me goes back to arguments that were highlighted by Bob Shulman, particularly over the last decade, where he really made a very simple point, but a very compelling one. And that is that if we look at brain energy consumption, expressed here as the as the cerebral metabolic rate for glucose in terms of glucose utilization, one sees first of all that there is, using any of a variety of measures, a relatively strong correlation across the brain between measures of neuronal activity, particularly synaptic activity, and glucose consumption. That's a very long described relationship. Sokolov, amongst others, was amongst the first to recognize it. Now, the second thing that became clear in animal studies and in human studies is that that levels of conscious awareness really are associated with very small differences in energy state near the top of this range. So when we apply anesthesia, we're moving along, we're suppressing metabolic and neuronal activity, but we're suppressing it really in this range up here for safe anesthesia. Induction involves relatively small changes in brain energy metabolism overall. Now, what we've learned from the brain resting state activity is that, of course, and from electrophysiological measurements preceding that, is that most of the electrical activity is occurring in this broad energy range. What we're seeing for the difference between anesthesia and conscious awareness is really a very small increase in signaling and a very small increase in energy utilization. So when we look at the task fMRI studies, what we're really looking at is modulations that occur in this energy band. The resting state activity is a reflection of the kinds of integrated brain functional activity that's occurring and using the bulk of the energy that we see. The notion of what we do as behaviors or individual perceptions as being modulations on a common framework, to me is an exciting way of thinking about the brain and about the way the brain has evolved. The first question is, why does the brain use so much energy to remain in an idling state? It uses this amount of energy to achieve a metastable configuration whereby very small, energetically small and kinetically rapid shifts can give rise to changes in perception or action, as you see here. So, a consequence of that thinking that has been tested is that the resting state activity includes information on the relevant states associated with task-directed activities, and therefore can express the variations in such states in its own variation. And one of the most interesting demonstrations of this came very recently with work from Saad Dubadi's work in Oxford, where he did something really rather beautiful. What he did is he took a set of resting state studies and on individuals who had associated task-related fMRI activities across a range of tasks. He used the task fMRI activities in his test data set to train a machine learning algorithm to effectively rederive the same information from the resting state activity using some simple filters. So what he's in effect doing, mathematically he used a slightly different technique, is he's training a set of lenses to search selectively in the resting state data of individuals for the underlying task-related activity across a variety of tasks. What he then was able to do is go to a second set of resting state activity from these same individuals using these trained filters and derive a predicted task-related activity for individual tasks. With a second set of task-related fMRI activities from those same individuals, he could then test his predictions and thus understand whether he could, in fact, see embodied within the information of the resting state all of the information on a whole range of tasks. Now, here we see illustrations of this across different subjects. Here for a language task, looking at the differences, actually it's a mixed task, differences between solving a mathematical puzzle and listening to a story. And what one sees here is in each case, the predicted map from the derived from the resting state data alone is shown in yellow. The actual tested map from the task is shown in red. And what you can begin to see as you look across each of these different individuals, across the different tasks, here is a theory of mind task. Here is a working memory two-back task. Here's a gambling task with a negative reinforcement. What you see, first of all, is that there is considerable overlap, in most cases, between clusters of thresholded activity in the task and the derived model in yellow. Secondly, what you can see is that if one goes from individual to individual, there are, in this very high quality functional MRI data from the Human Connectome project, there are reproducible differences between different subjects. This is the individual variation that really motivated the talk. And although the prediction ability of the algorithm varied over the tasks, what one can see is that in general, there was a great deal of the individual variation that could be predicted by the model. If you look, for example, here at regions in the parietal cortex but across individuals, you see the common localization, but you see here, for example, how the prediction has pulled out a little bit of this creeping, more anterior activation that you see here. They're really fascinating to look at. So it's given, I think this shows us that in principle we can derive much of that common information. Now this has actually led to, associated with this, there have been a number of direct attempts to sample individual variation, or to understand how to sample the individual variation within the resting state itself, and use that as a way of codifying distinct regions of the cortex in order to begin to understand variation in cortical functional architecture across larger populations. One of the first of these was by Wang and his colleagues at Yale, in which they performed what, and this is worth walking through once because variants of this have been used. I'll talk about ways in which variants of this same approach have been used in later studies. A base template was defined to give some prior information about the belief in the functional architecture of the brain. They used quite a loose description that of what they thought functional parcellation of the cortex might look like. They projected this into an individual brain space and then regressed this against the functional variation in the bold signal by looking, in this case, at correlational maps at a large correlational matrix relating geodesically long distance connectivities to seed regions in each of these template regions. And by iterating back and forth and reassigning weights in a way that I won't describe now, they were able to define functional architecture based on long-distance connectivity that could express a considerable amount of individual variation, as looking at across subjects here, yet preserve a common functional architecture within an individual across multiple days. Let's just take a look at this to begin to understand how this functional architecture expresses something of the characteristics that we would like to see in a meaningful parcellation related to human variance. Here, for example, if one looks at parcellation of the prefrontal cortical region, ignoring the arbitrary choices of colors, what one sees here moving across these three individuals is quite different parcellations of these relatively crudely defined frontal regions motivated by their crudely defined frontal regions motivated by their template. That's showing us elements of the individual variation. Yet if we look across from day to day within any individual region, say this light pink, you see it's adumbrated really very well. Many of these regions shown here as high confidence networks, of which this is one, really are highly preserved across the space. So, and with this, they were then able to demonstrate that they could, with reason based on the long-distance connectional architecture and this relatively crude template, they were able to assign identities to previously on a previously untrained data set for these individuals to the large majority of subjects. In other words, they could use this as the beginning of a fingerprint of the brain, in which each one of these described the fingerprint. And as you can see, if you know this architecture belongs to subject one, you can find subject one on, from independently acquired data or others. Now, if we can do this, we have a tool that tells us something about the way in which the brain is functioning. Although it's a highly filtered version of this, it tells us something about the way, and it tells us something about the ways in which the brains of different individuals may function in distinguishable ways. Now, can one begin to relate these differences to phenotypically or behaviorally meaningful variation in the population? This is, of course, at the root of the motivation set out by that slide that I showed you from Ed Bullmore to begin to map disease or to antecedents. Now, in one of the at this point, I'll tell you, I'll share with you that the answer is not is not well solved. I'm not sure that at this spatial resolution it will be possible, but there's still much to learn. And there are certainly some encouraging, modestly encouraging pointers along the way. Now, one of the first of these, although it was, to my mind, more disappointing than encouraging, came from Steve Smith's recent demonstration with the human connectome data, that an ICA parcellated connectional architecture of the brain, again looking at independent, spatially temporally independent varying components of the brain and their connectivities, could be used to express significant associations with a cluster of phenotypic measures on individuals. They defined, I'll show you the phenotypic cluster shortly, but they performed a PCA, a reduction of a whole variety of measures of individual phenotype, and found two large components whose variance across the population could be explained by the variance in brain activity after appropriate statistical correction. With these, here you see the functional relations between individual components of the architecture. Here you see the summary measures, which are basically reduced to one positive mode of variation, shown in red or orange here, one negative mode of variation. So there's a simple tray, which I'll show you in a moment, which is varying in one of two ways. And that was exciting, but the disappointing thing is the uninformativeness of the simple tray. What they found was what they summarized as a positive mode of phenotypic variation, which is attention, scores on a variety of tasks, including fluid intelligence, and a negative mode of variation, which was associated with problems in cognition, drug use, and a variety of others. Now, it's worth thinking about this in this context, because this map really goes straight back to Reichel, and we're looking at I think, an attentional map of the brain. So this is telling us something about phenotypic variation, but it's highly nonspecific because it's not giving us a clue to the individual architectures. Now, again, in an extension of this that was somewhat more directed, Todd Constable's group attempted to extract from the resting state activity an explicitly attentionally related network across a population, and then relate this to phenotype. So what he did is used a data-driven approach in which the phenotype drove the definition of connectivities on a training set, which could then be used for tests in an independent data set. What they found is that they could explain in their training set the relation. They could explain the functional phenotype with an R of about 0.8 that had a lower, as expected, but significant predictive capability on an independent data set. Now, we've attempted to extend this, and I'll go over this rather quickly, but it's because it's work in progress. But I think it's beginning to show us, to teach us more. There are two problems that we have with the previous approaches that are obvious. The first is the degree of resolution. I missed the discussions yesterday, but I can guess from the titles that, of course, what one is talking about in most instances, whether one is talking about recognition of action, faces, a variety of other features, are much, much smaller regions of brain. Functional parcellations of the brain at the level of individual phenotypic behavior is much, much finer than we've seen thus far. Secondly, the definitions of functional architecture have been highly dependent on prior beliefs about meaningful cytoarchitectonic structures. And so the more we can update these with more refined approaches, and the higher the resolution that we can begin to achieve, the closer we might begin to move to some aspects of this. Now, in one approach to this, we've attempted to move back to the local cortical architecture and not look at the geodesically long-distance connectivities, which are effectively double low-pass filtered, but simply employ this strong correlation that I showed you from Abkarian's work to look at individual architecture. And this, at least, has proven to be to give us highly specific views of the cortex. So here we show what these maps are derived. These are parcellations that are based on, in this case, an earlier version of the human connectome map with 150 cortical parcellations as the prior belief template, and these were remapped. The borders were remapped using a local K-clustering technique to look at functionally correlated regions along individual areas. This gives rise to near perfect assortment in test-retest data sets for individual variation. And as before, the regions show considerable inter-individual variation. Now, I'm not going to talk about that just for time, except to extend it to the more recent work where we have more redefined the prior belief template based on the Glasser parcellation in Nature earlier this summer, which integrates for the first time a broad range of structural data with prior cytoarchitectonic data and is slightly modified by task-related fMRI data. Using these 180 parcels for the brain, we were able to applied the K-means clustering method, and first showed that in independent data sets, once one defines an individual's cluster-based fingerprint based on the functional architectural modification of the template, that is to say, each areal region is redefined in functional terms, only 1 out of 820 subjects in the human connectome data set could not be accurately classified. The classification is 99.9% accurate. But more interesting is that, again, we see that that each of the parcels contributes, that we see here, contributes variably to the discrimination between individuals in the final data set. And those parcels that are colored in bright green really contribute effectively nothing to the individual variation. Those parcels that are in bright red express the variance that is most responsible for the individuation of the brain data sets. And I think this is a much longer story, but what I want to highlight particularly is you see these regions of high variance are in precisely those areas of more recent heteromodal cortex, and particularly in this very interesting region of the cingulate. Now, part of this expression of variance is related to what happens in what changes in an individual from day to day. The functional architecture has a state as well as a tray dependence, but again, one can begin to correct for this by taking resting state images on multiple days, looking at the within subject variance and expressing now a map of the functional individuation across parcels as a normalized measure of the between subject the between subject difference with respect to the individual subject difference normalized to their total variance. And here again, one sees areas of the frontal and temporal cortex contributing most to this individuation. Asking the same sorts of questions that we started the talk with about the contribution of genetic heritability to this, we see that some of this variance is heritable, relatively heritable. And in looking at monozygotic versus dizygotic twins versus unrelated subjects, we see that the parcellation is progressively more dissimilar as we move to unrelated individuals. And here you see those parcels that show high relatedness in monozygotic individuals relative to the others. Well, I'm going to close now, except to point out that where we really want to go is to begin to understand the way in which this individual variation is modified by environment and lifestyle. We're beginning to get a handle on some elements of it. What we really the importance of trying to do this at this scale, despite its limitations, is that we can move to large populations. If we want to look in a cohort study design, for example, for lifestyle interactions with any variants, such as I've described before, we really need to get up to tens of thousands. It's a much longer story than I have time for now, but within the UK Biobank project, we've now set out firmly on the path towards imaging 100,000 people to begin to provide the data sets that will allow us to make this sort of variance. And the very first taster, not of results, but of the kind of data that can be acquired from this, we published last week in Nature. And it's what I want to just emphasize from the first 7,000 of these subjects is while there is a great deal to be worked out, this is going back to a simple functional connectional architecture and its relationship across a very wide range of variables, phenotypic variables related to physical activity and lifestyle, to cognitive phenotype and so on. What I want to express is not any individual result, but even doing massive, very crude Bonferroni corrections across all of these associations, one begins to see that there's a great deal of potentially meaningful variance that can be associated across these populations, giving us some confidence in continuing to explore the way in which we can move across scales, particularly exploring this mid-ground between meso and macro scale architectures of the brain and behavioral differences in trying to reconceptualize a human disease and its antecedents. So let me stop there and thank you for your attention. I'm impressed that you're more awake than I felt only an hour ago. Thank you.
And we have to make a remark here, and apologies if I take 1 minute from the discussion time. Paul and I have been discussing very heavily the future activities of our new cognitive neuroimaging center here on the campus, which is just ready to start its operations. And in this context, as well as in the context of the present conference, namely grand challenges in brain cognition and good life research, this talk has an important role, because what we would like to do here in Singapore at NTU in our new cognitive neuroimaging center, and within the present context, can be found a real scientific basis in Paul's talk. Namely, we would like to join with our activities of the UK Biobank Neuroimaging Enhancement Study with a Singaporean chapter, using the very same protocols in the Singaporean, Chinese, Malay, and Indian populations, and contribute with exactly the same protocols, same type of scans to this huge enhancement database, which you call the imaging enhancement database, 100,000 Caucasian UK subjects, brain fingerprint, individual brain fingerprint, and adding to it with the local Singaporean, Chinese, Malay, and Indian individual brain fingerprints. But one more important aspect was added to this whole picture by Paul recently, namely and this is related to the essence, the context of the present conference, some kind of individual variation or maybe ethnic group related variations in good life aspects. You mentioned in one of your last slides this sort of positive, negative attitudes, what is acceptable, what is enjoyable, what is good life aspect, and what are the negative aspects in life. So all these things are now, at least certain aspects of these things can be rooted, can be anchored to or rooted in Paul's present lecture. So Paul, thank you so much for this guidance related to our future activities. Of course, you are part of the team, so it's not, and we have been discussing it for so long in the past. And I also would like to express my thanks to Jan, my sparring partner over there, because this conference gives a fantastic impetus to the future activities here on this campus regarding cognitive neuroimaging and other aspects related to grand challenges in brain research, cognitive research, and good life research. So now, sorry for this very short sort of conceptual background giving. Questions? Guy, you had a question.
It's sort of a long-winding comment finishing in a question. I've always been very much disturbed by the so-called functional parcellations that make no sense to me, because in certain, I mean, the difficulty is that we don't really know what the parcels are in the human brain, or to a large degree. There is one bit of cortex where we have a better guess, because there's retinotopic organization, which we have mapped in great detail. There are 19 areas that are now published. David and Matthew were actually part of that effort, so they know it. They incorporated it partially in their recent Nature paper. They took most of the areas. So the general point is that you really need to try to get to a proper definition of the functional. And the difficulty I have with the recent Nature paper is that the parcellation in IPL and in parietal cortex in general makes very little sense. It's way too crude. If you read some of our recent papers, we even subdivided the cytoarchitectonic regions of Zille's group. So there is a much finer functional and I think areal scale than what you have, which would, and that leads to my question. The one part that really stuck out at the end that was green is the parietal cortex. And is that because it is stable, or because the parcellation actually is way too crude?
No, I think you're asking very very germane questions. First, I think we need. My own view is that parcellations are they're way stations, and they to be useful, they need to have a purpose. And some will be more useful and some will be less useful. There is no, and in fact, to be fair, Glasser and David both suggested in the Nature paper that this was an interim definition of the way the brain might work. And it has some cytoarchitectonic basis, and obviously that varies from region to region. I think what to me is exciting about the approach integrating imaging methods is it gives us much greater flexibility to look at behavioral, phenotypically meaningful parcels, at least in principle. Now, the negative side is of course, we're still struggling with resolution. There is no question in my mind but that a much finer grained parcellation is going to be needed if we want to explain a great deal. And I certainly agree with you that that one of the reasons for failure of regions to individuate is that we've selected them imperfectly and thus average signal that shouldn't be averaged. Now, there's a lot that one could say, but I think the next step in this is the Glasser-Van Essen paper, to my mind, was a tour de force in integration of data. But at the same time, as an integration and data effort, it's limited by the fact that it is basically hearkening back to a past and a conception of the brain which carries strong biases. I think it in principle remains possible to develop much more data-driven and pragmatic redefinitions of parcellation. And if we can get fine enough grain to it, those might provide the refinement of these kinds of maps that will become progressively more useful. It's just I think it's just saying what you said all over again, Guy.
Yeah, thank you.
So here. So first of all, thank you very much for this comprehensive overview. I'm not a neurobiologist. I come in from microbes, how they impact body physiology. But for me, when I was listening to you, you made a point, which is my first comment, is on schizophrenia. And you made a point that there is a reduced connectivity. You were hesitant to say it, but you sort of raised the issue. And one aspect of schizophrenia is, of course, reduced access to nutrients. So would you then dare to say that in a window, let's say 4 to 7 years of age, where there's an enormous shunting of energy to the brain, that you may be exposed to reduced connectivity if you have a malnutrition condition? This is my first question. And the second one is related to the architecture that you show in the end, which of course is very crude, but still is an embryo of a systems biology approach. So if you now take extreme conditions like voluntary fasting, would you change the patterning? Or for that matter, if you go into pregnancy, which is an enormous change of energy consumption, and you actually, which I really liked, you're the first one to use the word metabolism here.
So, Sven, I think you've, I don't have answers to any of those questions. But what I would say is that what is important clinically and what is important for us as a society is to find, is not to, we can't change the genetic architecture of our population by ethical means, but what we can do is change the lifestyle and environment and nutrition that we expose it to. So I think these methods should potentially allow us to look at the interactions between nutrition and these measures of connectivity in ways that we might be able to infer some useful outcomes. In terms of beginning to look across lifestyle too, really exciting things are starting now in parallel with the adult connectome project is of course a neonatal program that David Edwards has been leading from Europe. And I think the hope is that with time we're going to be able to first stitch together these populations and secondly longitudinally image developing populations at such scale that we can begin to look at some of the questions you've raised about development more directly, following along the lines that Peterson and St. Louis first highlighted a few years ago.
Well, I have a rather polemic remark to make. I can read you from the work of Glasser and Vanessa. We characterize 97 new areas and 83 areas previously reported using post-mortem microscopy or other approaches. So now we have 170 areas. If you look at this area, this one for gambling, for example, which is placed in the motor cortex. Well, it's serious science, or is it just a mixture of two type of approaches? One is cytoarchitectonics, immunoarchitectonics, which is very serious, and the other is based on bold, which of course it's not so precise and can so modify the cytoarchitectonics, saying that since there are more blood in one part of premotor cortex, that's the gambling area. So it's really difficult to accept this paper as really foundation for something new, as they claim. It seems to be rather a mixed for political reason, because a lot of money has been invested.
Okay, well, of course, I wasn't involved with the Glasser paper, so I have no vested interest. I think it's very dangerous to suggest that a scientist is doing something just for political reasons. I think this is a continuation but what I would, as I even stated at the time, I think these are these are pragmatic maps. The whole idea of a map is to distinguish differences. Now, what you're really raising is not the question of whether those differences can be found in different regions, but whether it is meaningful to group them together as if they had equivalent, as if they could be interpreted in an equivalent way. And I think that's a really fair comment. I think one of the problems with the cytoarchitectonic maps, as you know from the work of Ammons and the work out of the Montreal group, is, of course, while the borders can be defined, it's very difficult to get up to any sort of scale. And so we actually have terribly limited information about individual variations in these patterns. And it's unlikely that that's going to change very rapidly. I think what Glasser would probably argue is that most of what went into the Glasser paper was not functional data, but structural data based on indirect measures of the microarchitecture of these areas as reflected in the water signal. Now, exactly what the correlates of those are is unclear. I, for example, am I found it very irritating that they kept talking about myelin maps. I mean, those aren't myelin maps. They're maps of a biophysical property. So I share some of your irritations. I think that if we can take an area that we presume to be cytoarchitectonically homogeneous, and we find that there is a functional variance within it that is reproducible, that does tell us that different parts of that map are distinct. Whether that distinctness is preserved is another question. I just come back, the final point I'd make is I think you have to be very careful too about trivializing the functional imaging too much. I don't think anyone would claim that there's a really gambling areas in the Muller
But they wrote that there is a gambling area.
What's that?
They wrote in the paper, it's area 55.
I know, but I don't think that,
55B.
but I think if you spoke to David Venison, he wouldn't say that's where gambling is. He would say that's an area that is distinctly activated, differentially activated during gambling, along with many other areas of the brain. And the distinction there is really important because it doesn't tell you that area does gambling. It tells you that there's something about that task that makes that area distinct. And that's why I'd just be a little bit careful in how you... And so all it's saying is that within a broader cytoarchitectonically defined area, there is a way of distinguishing one section as, at least for an interim map, as being different. But I share your reservations. I mean, these maps are what they are, and that's why, I think the biggest challenge for us is to find much more unbiased ways of approaching them.
Well, to final comment first, I noticed that you also smiled when I mentioned the gambling area. And the second point, why the gambling area should be in the motor cortex.
Seems like a pretty good place to me.
Apologies, but we have to close this discussion. There was still one last question from Peter, and the rest, Stan, sorry, should be for the coffee break.
This exchange just shows that scientists are only human, so whether they do things for money or not is debatable. But more seriously, you emphasized individual variability and heritability of cortical structural maps and cortical activity maps. Now, we know from our parents that the rest of our body just as much heritable, not only the cortex. And there is just as much, if not more, individual variability in body plans than in cortex. If you or others look at the subcortical structures, particularly directly related to brain, such as thalamus, basal cortex, directly related to cortex, such as thalamus, basal ganglia, and basal forebrain, are they more or less variable than the cortex.
Yeah. Peter, as usual, an astute question. I don't know the answer to that. I think we have much less information. I think part of the challenge has been that the meaningful parcellation with these methods has been much more limited, predominantly because of spatial resolution. We're also talking about, when we talk about variance, of course, in looking at the cortex in isolation, the neocortex particularly in isolation, we're looking at just a fraction of the brain. And there is, my own belief is that the future for this also is in moving away from, I don't know how to visualize this or represent this, but moving away from these kinds of traditional maps, which we can instantiate in something that we can relate to a physical surface, to something much more all-encompassing, in which we're looking at the temporal dynamics of these interactions as well. All of this is compressed into a static framework, and I don't need to tell this audience that that's it takes away 90% of neuroscience if we looked at static frameworks. So I think you're right. The subcortical structure is hugely important for their interactions. They must show variation. We simply don't have a handle on it. The second thing is that the temporal dynamics, we're pretending as though it doesn't exist, except as summarized in a mean correlation. And these have got to be solved in the future.
Apologies to those who couldn't raise a question now, but please ask Paul during the coffee break, because in the interest of time we have to start our coffee break. We will convene again here at 10:45. And one more time, Paul, thank you for your fantastic lecture.